All right, everyone. Thanks for joining us for this week's seminar. This week, of course, we have our very own Dr. Frank Carroll presenting on Bayesian clinical trial designs. Dr. Harrell clearly needs no introduction, but I'll still introduce you. Of course, is one of our own faculty members. Received his PhD from UNC, joined Vanderbilt back in 2003 when he's our founding chair up until 2017. Frank served within the US FDA, holds a role of expert biostatistics advisor at the Center for Drug Evaluation and Research, and was expert biostatistics advisor to his Office of Biostatistics from 2016 to 2020. He's an associate editor of Statistics Medicine and a fellow of the ASA. He also won his W.J. Dixon Award for Excellence in Statistical Consulting in 2014. Frank's specialties include development of accurate prognostic and diagnostic models, model validation, clinical trials, observational clinical research, cardiovascular research, technology evaluation, pharmaceutical safety, Bayesian methods, quantifying predictive accuracy, missing data imputation, and statistical graphics and reporting. And of course, Frank maintains a very active presence in online communities. And like I said, he'll talk today about flexible Bayesian clinical trial designs. Frank, the floor is yours.
Thanks for the nice intro, Siwan. And I want to encourage people to ask questions while I'm talking. I love interruptions. There'll be plenty of controversy in this topic. And I hope, will you be watching online to see if somebody asks something in the
Until 1:40 for this, so folks definitely feel free to have the department meeting at 1:45, so that'll be time. 2:45. 2:45, sorry.
Yeah.
Sorry, 2:45.
Yeah.
Yes. Thank you.
Okay, so there's a link here to these notes, these slides, and there's a link to the detailed chapter in my big Bayesian course notes where you see the code for everything I'm going to be showing you and a lot more elaboration, a lot more details. So I want to get right into what are the challenges in doing clinical trials. There's lots of challenges, and the first thing that goes wrong in a clinical trial is not being able to recruit enough patients. I can't really solve that problem. But the second most common problem in a clinical trial is you get through and your result is equivocal. So in the frequentist world, equivocal result means a p is greater than 0.05. Fisher said, what you do when p is greater than 0.05 is you get more data. That's the only conclusion. Now, if your confidence interval is very narrow, you might know more than that implies. But your worst thing to happen would be a wide confidence interval and a p is greater than 0.05. So you can't reject the null hypothesis and you don't know anything except the money was spent. This is my estimate based on simulating several clinical trial scenarios. Since we have so many trials ending in an equivocal result, it's a real shame because if that's the result, you could have made the same conclusion about 1/3 of the way through. So if you were doing aggressive futility stopping, you could have stopped early for futility with the same amount of knowledge that you had waiting for the whole study to finish, which is nothing. So I'm going to be making a strong argument for very aggressive futility stopping. Sample size estimates are notoriously inaccurate. Every statistician will tell you it's the most distasteful thing we're ever asked to do. And people have called it the sample size samba. It's just a dance, it's a game, it's very distasteful. Now, the problems are very serious for all clinical trials. Once you get into rare disease where it's hard to recruit enough patients, things are more acute. Futile studies tie up patients for years. If there's no available treatment and you're using hopefully the first effective treatment in a rare fatal disease or a neurodegenerative disease like ALS, a trial that's going to end up being futile is going to tie up patients for 2, 3, and 4 years, sometimes 5 years. Wouldn't it make more sense to let those patients be turned loose to be in other studies instead of tying them up for a study that's going to show no effectiveness, or it's just going to be equivocal. Likewise, you should be able to extend a study that's promising but not definitive. So what are the culprits? So one of the culprits is alpha spending. Frequentists or traditional statisticians worship at the altar of alpha. And the degree of worship is so scary to me. It's like a fundamentalist religion. And it's based on a false belief. And that's especially hurtful when you worship something and it's based on a false belief. So the false belief is being that you're controlling the probability of being wrong. And that's not what alpha does. So alpha is an assertion trigger probability. It has nothing to do with the probability of making an error. So we call it an error probability. That's where the misunderstanding started. And that's one thing that makes people think alpha is blessed in a certain way. I'm going to argue that alpha is not even a noble cause. It's so unrelated to the probability of an error that it's amazing that we use the word error when we say type 1 error. Another problem is thinking of the sample size as a constant. It's the most random of anything we do. The sample size is a true random variable. And then what about the minimum clinically important difference that you use in a power calculation? We pretend that's a constant. We pretend that we know the constant. And then if the cost of the study is too high, we increase the MCID so that we can do the study on the cost that we were given to live under. So that's the whole game playing that we get into. Another big culprit is a low information outcome variable. So these are goals and desired conclusions of a randomized trial. This lays out 7 different goals. You don't need to have all of these goals, but the one that we tend to concentrate on is finding the effectiveness of the treatment. I'm going to suggest a very good additional goal is to demonstrate the amount of efficacy is greater than trivial. So we need to finally address the mismatch between clinical significance and statistical significance. Another conclusion might be inefficacy. Right now, if P is greater than 0.05, you cannot conclude that the treatment doesn't work. That's exactly not what it means to not reject the null hypothesis. With a Bayesian design, you can actually make a conclusion that the treatment doesn't work. So remember, in the p-value world, you're assembling evidence to reject something. You never assemble evidence in favor of something. We might also have a conclusion of harm, non-inferiority, similarity, or we might just want to estimate the treatment effectiveness without actually saying, are we concluding that's effective or not, or harmful or not? So those are all valid goals or conclusions you might want to draw. And the thing we want to avoid almost at all costs is to not have any conclusion when you get to your planned maximum sample size. This is the worst thing, because that means you have enrolled patients, you've made them experience sometimes risk, sometimes just a lot of their time, as well as the investigators, and you don't know anything. We really don't want that to happen. We're going to work very hard on that one. And then we have to talk about what are the desired operating characteristics of a design. We've been given a certain way of thinking about alpha and beta. Those were never a good idea for the design. Beta maybe is more okay than alpha, but the real operating characteristic that we need to care about is the correctness of the conclusions. That's very basic, very direct. What do we mean by correctness? What I'm talking about is the accuracy of the conclusion or the probability that your conclusion is the right conclusion. That's what I mean by correctness. We also want to have good sensitivity to detect a very effective treatment. So if the treatment effect were actually at the MCID and you had no information to be able to find that or detect that benefit, that would mean you have low power. So whether you're talking about Bayesian power or frequentist power, it needs to be a decent level of power. This is another operating characteristic that we're not taught to emphasize in the frequentist world, which is your expected stopping time. We don't really give credit for being more efficient than the sample size you planned on. So I think we should give credit for that. Can we lessen this? We want to get adequate precision if there is evidence for effectiveness. So one of the characteristics of the design I'm going to talk about is that if we have evidence that the treatment is ineffective, we don't care to estimate how ineffective it is. But if we have evidence that it's effective, we want to have the ability to estimate how effective it is. So that's explicitly stated here. We need to understand, these are challenges now. Big misconception here that if you have a Bayesian design, you need to calculate alpha. So if somebody tells you you need to calculate alpha, you tell them you can really simplify this project by getting rid of me and not having me involved with this project. You need to go with something basic, because if you're interested in alpha, that means you're not interested in the accuracy of your conclusions. You're interested in something that is a statistician's friend that's not a clinician's friend. The correctness of the conclusion is the only relevant operating characteristic for post-data decision-making. When you calculate alpha, it's not a function of any data. Alpha is a pre-studied tendency to trigger an assertion of an effect if there's no effect. So alpha is not a function of data. So it can't really tell us anything interesting about the performance of our decision process. And so we need to use something that's relevant to decision-making based on data, not based on pre-studied tendencies. We need to quit allowing gaming of the effect size used to calculate the sample size. We need to be willing to stop very early for futility. We need to decide whether we want to allow ourselves to stop early for similarity, with a strong conclusion that the outcomes are similar regardless of the treatment. We need to formalize a minimum precision criterion. We need to be specific and objective about triviality of treatment effect, getting into clinical significance. So those are the challenges. So now, what are the features of the different paradigms? The frequentist paradigm is usually using p-values. The operating characteristics everybody seems to care about are really strange conditional probabilities. They're conditioning on unknowable quantities, which is a very strange thing to condition on. And so what you're asking yourself is what may happen under an unknowable assumption about the treatment effect that generates the data. So when you're calculating things like power and alpha, those calculations are using unobservables. They're using things you can't possibly ever know, such as the true treatment effect being zero. Alpha is not the probability of making an error. It's merely the probability of making an assertion about effectiveness given some unobservable quantity. Alpha does not condition on the data, and that makes it have multiplicity problems. Because when you calculate alpha, alpha is a function of the looks that you intend to make on the data. It doesn't
Frank,
matter if you actually take those looks or not. It's a function of the intention to look at the data.
hey Frank, I have a question.
Go ahead.
I appreciate your point that you're conditioning on an unobservable, and that makes it sort of a strange thing to talk about under that way of thinking about it. But what that unobservable thing isn't just any unobservable thing. The unobservable is specifically the opposite of the assertion that you're making. Is that not good enough reason to conceptualize it as an error? Even if it's not the error that you would necessarily care about, and I get that and appreciate and respect that, it still seems like a reasonable use of the word error to me of that unobservable thing as the opposite of the assertion you're making.
I can halfway see your point, Andrew, but if you looked at anybody in the world who is not doing frequentist statistics, what is their definition of an error? They would use the word, along with error, they'd use the word mistake. And when they talk about a probability of making an error, they would say the probability of making a mistake. Now, it's only in frequentist statistics that you not use that definition. That's the only place I can think of in the world. So anyone else, if you're betting on a basketball game, you're betting on a horse, you're deciding whether to cross a busy street with traffic, you are not conditioning on something that's unknown. The way you conceptualize risk and what can go wrong, what's your probability of being hit by a car? What's your probability of being wrong in the decision that you make? That is something that is an unconditional probability in the sense that we're talking about now. So I think the use of the word error in the way that you would like it to be used is only a definition that would fly in frequentist statistics and not in the rest of the world. So I would love to continue that discussion at more length at another time. So in Bayes, this is where multiplicity problems come from in the frequentist world, is how many opportunities did you give data to be extreme? And the opportunities come from the number of intended looks that you're making at the data. Bayes doesn't operate that way. So it makes decisions based on posterior probability that a clinical assertion happens to be correct. And the operating characteristic is the correctness of the decision. And the correctness of the decision is not going to really use unobservables. So this is the shortest way to say, how would you compare the two? In the frequentist world, you're talking about the chance of getting a positive result when there is nothing. So when the treatment is ignorable, it can't hurt people, it can't help people, it's totally ignorable. So what's the chance that a positive result occurs when there is nothing? Bayes is about the chance that a positive result turns out to be nothing. So what's the probability if you make a decision that the drug works, what's the probability that it doesn't work? That's exactly what Bayes is concerned with. So I hope these words here help frame how drastically different, night and day difference, the two paradigms are. So more about Bayesian operating characteristics. In terms of the positive direction, if you say that the probability is 0.97 that the treatment works, and you want to act as if it does, you approve the drug, the chance that you're wrong is 0.03 if the prior used to calculate the 0.97 is the same as what some critic would use who has a lot of power and a lot of say. So if those two priors agree with each other, then you'll be having to both of these people who specify this prior, your probability of being wrong is 0.03 if the probability of efficacy is 0.97. The Bayesian power is dominated by the prior, is affected by the prior, but is dominated by the sample size and the uncertainty in the minimum clinically important difference. And so the only interesting simulation to do, the only interesting Bayesian operating characteristic to calculate is what happens to the probability of your decision being correct when there's a disagreement in the priors. So that's where we need to spend all of our effort. If you don't have some sort of really strong agreement between the investigators and the reviewers, if they totally agree on the prior, then you don't need to do what I'm about to say. But if they disagree, you need to show that the prior that you are going to use in the analysis doesn't cause some major conflict down the road. So that's what we're going to simulate. So we might simulate 1,000 randomized trials with 1,000 different values of the true treatment effect. Our goal with Bayes is to uncover hidden truths, relying only on observables. So of those 1,000 trials in which a certain decision was made, we count how often the decision was correct. So this is where you have to be crystal clear on what's the numerator and what's the denominator. You subset the trials to those in which the decision was made. the decision might be, we're going to act as if the efficacy is positive. Capital delta is greater than 0. Now we reveal the delta that generated the data in those trials and check how often the delta was in the interval, such as delta greater than 0. How often was it positive? So we're just asking how often we were correct in what generated the data.
Interval, you just mean the portion of, say, the number line. You're not talking about a confidence interval.
Right, it's the portion of the number line, real number line, yeah. Efficacy greater than 0, efficacy greater than 10%. Harm would be like less than 0. Similarity would be in a small interval around 0. Whatever interval you care about. So it's a post-data consideration, is conditional on the data and the decision. It's never conditional on capital delta. So this maybe is a better way to frame this friendly argument Andrew and I are having. What's the best way to measure accuracy that is useful in judging how decisions are made? And I'm just claiming that besides any philosophical argument, when I calculate accuracy, I don't want to make an assumption about this that I could never know is true or not. So just like when you're doing predictive accuracy in a prognostic model, clinical prediction model, you're seeing whether your predicted probability matches like observed proportions of death. So simulations reveal how the differences in the paradigms are dramatic. In the frequentist simulation, you might simulate 1,000 trials in which the treatment is irrelevant and compute a test statistic and count how often P is less than 0.05. That would be your alpha simulation. Likewise, for power, you might pick an arbitrary positive treatment effect, pretend it's a single number, and find out how often you can get a p-value less than 0.05 when the effect is that number. You have multiplicities because you're calculating the probabilities of data being extreme, and you're not calculating probabilities about delta. So now we get into the sequential design part of this. I'm going to submit that sample size calculations are truly voodoo. They're truly a total mess, a total embarrassment to statisticians. And if clinicians knew how silly some of the things they do are, such as an investigator came to my office in my previous institution and said, I want to detect a 25% reduction in some physiologic parameter in a mice experiment. And I did the calculation. She needed 20 mice. She found out how much that would cost. Each mouse was like $10,000. These were very special mice. And she said, I can't afford it, but I made a mistake when I talked to you the first time. I want to be able to detect a 100% reduction. And I said to her, you know, if that's true, you probably only need 4 mice in your experiments. But that's the kind of game that I have seen played out through my career. And the MCID is gamed. It really has a lot of uncertainty, even when you're not playing the budget game. Majority of randomized trials are equivocal. Some of that is because of playing this game with the MCID. Could have stopped much earlier. And so here's a quiz for the group. If the current treatment effect is going in the wrong direction, how far along in the study can you be for you to still have a good chance, like 0.9 or greater, of ultimately showing that the treatment works in the right direction? So how far along in the study from 0 to 100%, if you've got the wrong direction of the treatment effect currently, that you can get a reversal and end up showing evidence for a benefit? We got a 20. What else we got? You got to be brave. There's no penalty here.
How harmful is harmful? If I'm saying something harmful, I mean.
Anything left of 0. I'm not saying how much. It's just on the wrong direction.
19.5.
Nah, that's Nah. Do we have any different, substantially different prediction?
60%.
60%. If you want to get drilled down, it does, but this calculation that I'm basing this on is just that it's the wrong direction. So the answer is 10%. So if you're more than 10% through the study and the treatment's not in the right direction, you have very little hope of having a successful study for benefit. And that has a lot to do with framing this current discussion.
Like you think 10% is worst case? Or
10% is a kind of stylization. What's the word I'm looking for?
Yeah.
It's a stylized calculation that David Spiegelhalter did. That's details are in the course notes in the link. So what makes Bayes really great for sequential looks is because you're calculating probabilities about delta. You can do infinitely many looks at the data and have zero multiplicity issue. So if that sounds like a disaster to you, just go through the chapter in the course notes about sequential, where I do a simulation with infinitely many data looks. Infinite really meant 500 in that example. And you can see that there's absolutely no problem with multiple looks in the Bayesian paradigm. And the reason for that is, there's a lot of ways to say it, but if you calculate a posterior probability that people would believe at the end of the study, you're going to use the same mechanism and the same prior for calculating probabilities at any point earlier than that. So if it's valid here, it has to be valid at any point in time. And the other thing is the posterior probabilities that you calculate early just become obsolete. So Bayes formally respects the flow of information. Frequentist world doesn't do that. Alpha is kind of bidirectional in information flow, but in the Bayesian, it's forward information flow. So an early look just becomes obsolete when you have a new posterior probability. The probability of making a decision error is independent of the stopping rule, and the expected sample size is minimized by maximizing the number of looks at the data. So now we're going to go through an actual design, and we're going to simulate how that performs to drive home these points and to show how you can make decisions, make accurate decisions, and you can make them a lot earlier.
Sorry.
Yeah, go ahead.
Like about that 10% thing that you just mentioned, like, does that mean that, that if I'm going through like my trial and I got 10% of my outcomes collected and realize that there is a negative, like the point estimate is negative, I can stop the trial? Is that the?
You probably should stop it for hopelessness.
Okay.
Yeah, the only way to not be hopeless is to make that 10% into 5% by doubling your sample size. So if you allow yourself to go much farther, running the risk that you might be wasting twice as much money, but it would give you a better probability of success. But that 10% is, relative to your total sample size. Yeah. So we have a two-group comparison on a continuous variable that's normally distributed, and the standard deviation is equal to 1 in both treatment groups. So this is like the simplest problem we ever have. Two sample problem, normality, equal variance, and known variance. So we have the goal is maximizing the probability of making the right decision. We're going to allow ourselves to have 4 conclusions. One of those is no conclusion, and the other ones are real conclusions. We want to minimize the probability of an inconclusive study result at the study end. We want to minimize the expected sample size, stop as early as possible if there's a high probability that the treatment effect is trivial or worse. Now, by adding trivial in here and not just 0, that allows for much earlier stopping. So what most futility calculations fail to recognize is that if you're not futile, but you keep the study going to the end, and you have a p-value of 0.049, and you have a 2 millimeter reduction of blood pressure that no clinician cares about, you can't call that much of a success. So we're going to recognize that we would like to have some evidence of something more than a trivial effect in our futility stopping rule. Stop early for non-trivial efficacy, and making sure that we don't stop too early, that we can't estimate how effective it is. This is a criticism of a lot of adaptive designs. This is not an adaptive design. This is just a sequential design. But a lot of adaptive designs, they choose the best treatment with a good probability. But if you ask how big an effect is it, they can't give you that. So we're trying to prevent that problem. We're not going to allow gaming. We're going to recognize that traditional sample sizes are arbitrary, recognize that what appears to be a study extension to one person will appear to be an interim analysis to another person, and that person may not even need to do a sample size calculation. You need to do a sample size calculation in the frequentist world because that gives you what alpha is spent against. In the Bayesian world, you don't have alpha to spend, so you don't actually need a sample size calculation. You're going to be a better scientist if you do not do a sample size calculation, but you're going to drive your budget people crazy. Build clinical significance into the quantification of evidence. So here's our symbols. Delta is the true effect. Little delta is the MCID. Gamma is the minimum detectable delta. So I have different words that I use, like triviality threshold, minimum detectable, treatment effect. And just to throw out an idea that what this would typically be would be half of the MCID or 1/3 of the MCID. That's just a good starting point, and then the clinicians really need to think that through. And then we have a similarity interval, which is plus or minus 1/2 of the MCID.
I have a basic question about similarity. So it's the delta plus and minus the triangle and.
Yes,
Okay.
thank you. I never caught that error. I've been saying this for a long time.
Oh,
Thank
so
you.
that covers zero means they're similar.
Yeah, it should be delta plus or minus.
Okay. So if they cover zero, they're similar. But what's the difference between similar and like non-conclusional?
Between similar, yeah, similar is a definite conclusion. So
Okay.
it's much different than a non-conclusion.
Okay.
Non-conclusion means we don't have evidence to put you along this whole line.
Okay. I guess I'm just thinking, what's the difference between this one and comparing to if a confidence interval covers zero?
What is different about this is in a confidence interval, you specify the confidence level and it gives you back the interval.
Okay.
And in the Bayesian world, you specify the interval and you get back the probability.
Okay.
So we want to have control over the interval because that's a clinical specification,
Yes.
whereas a confidence interval will get you back a statistical specification.
Can I ask another question?
Yeah,
The minimum detectable delta, is that a property of your outcome measure, or you're saying that's a?
yeah,
property-
which we'll get to.
Okay.
Good point. All right. We need a sample size that might be calculated assuming delta is equal to little delta. We're going to calculate this sample size, this largest sort of sample size, to achieve a Bayesian power of 0.9. In other words, the probability is at least 0.9 that you will get a posterior probability greater than 0.95. So Bayesian power is much simpler than frequentist power because everything is like in the predictive space. Whereas the frequentist world looks like a retrospective space. So everything here is actually simpler. So you want to have a high probability of getting a high probability of efficacy if you're at the MCID. Now, we could also say, and what I'm going to do is I want for my evidence, I'm going to use a decision criteria that says we have a very high probability that the treatment is working in the right direction, and we have a pretty good probability that it's more than trivially effective. So I have a dual criteria, and I need to satisfy both of these. And then I'm going to define N sub P as the minimum sample size for the first efficacy assessment. This is going to be based on the width of the posterior distribution. Like in the frequentist world, the width of a confidence interval. So I need that width to be small enough that I know something about the clinical effect of the treatment. There's no separate futility analysis because inefficacy is a easier thing to conceptualize and it subsumes futility. So if we have evidence for inefficacy, you know, that has as part of that what people would call futility. But to me, it's an easier concept. And so getting to Lori's question, we're going to be talking about the resolution of the dependent variable, and you can make that resolution higher by breaking ties or measuring it as a longitudinal repeated measures outcome or extending the length of the follow-up. Those are the three ways we get higher resolution outcome measures. So this is what we start with. Choose an outcome variable with the maximum resolution that's relevant to the patient. That's how you get maximum Bayesian power or frequentness. We elicit the effect sizes delta and gamma for this Y from the experts and patients who are unaware of budget limits. Now, this unaware is all important. We can't play this game anymore and expect to not have the problems we always have. The way I describe it in a nutshell, if you power a study to detect a miracle and the only thing you get is MCID, you're left with nothing. We just can't stand that anymore. We need to be very loud about this. Okay, you compute the expected sample size or the maximum sample size, perhaps, and the minimum efficacy sample size. Are those sample sizes feasible? If they're not, can a higher resolution outcome be developed? This is your only recourse. Your recourse is not to change the MCID because then you make everything a dishonest game. Your only recourse is to change the dependent variable or to change the design totally. So if you change to a crossover design, you know, you'll have a lot greater power in a crossover design than you do in a parallel group design, depending on your feasibility of the treatment. So if you can't develop a higher resolution Y variable, you should quit, not enter into the game. But if you can, you go back up here and now you elicit the effect sizes and start the process over. So now if you get a feasible N, because now you've got 6 visits per patient and you've gone from a 3-level outcome to a 20-level outcome measured 6 times, now we can do it without having too many patients. And we begin the study. And
pilot study and then you use it as your prior for your next study. So you don't have enough power to do. You're saying just don't even start the trial.
yeah, a pilot study, you know, there's a lot of discussion about what a pilot study is good for and not. And a lot of people say it's mainly good for estimating variability, not for much else. So it also depends on what you mean by pilot study, but there's these things called like phase 2, 3 bridging studies. Those come into play and can have some nice features. So we asked the question, do we have a very low probability of more than trivial efficacy? If that probability is very low, we can stop early for inefficacy, and we have a very low chance of a non-trivial benefit of the drug. If that is not very tiny, have we followed at least N sub P participants so we can estimate the precision of the treatment effect, and it's a good precision? If no, we're going to accrue more patients. And if we're up to this point now, we can look at our evidence. We have strong evidence for efficacy, and we have pretty strong evidence for greater than trivial efficacy. This might be MCID over 3. Now, what about, is that met? Well, if that's met, you stop with a conclusion of non-trivial efficacy. If that is not met, you can look to see if you have evidence for similarity. And if you don't, and your resources are not exhausted, you keep going. If they're exhausted, at this point, you stop with the inability to reach any conclusion. This is our worst outcome other than the treatment killing patients left and right. We could stop early for similarity. So you see that there's a definite order of things that you might look at, and we wait to look at the evidence for efficacy. We wait just a little bit. So that is the design. And you can see that it's very goal-oriented because you have multiple goals and precision, finding evidence for inefficacy, efficacy, similarity, and this design will meet those goals. So let's simulate the performance. So with this 2-group problem with normal variance 1, the frequentist sample size for the N is 234 for getting a power of 0.9. Now the Bayesian power is going to be larger because Bayesian assessments are directional. This is a little bit nitpicky, but you can get a great Bayesian power for this directed assertion using 196. But if you wanted to bring in the evidence for more than trivial effect, and if we defined the MCID as 0.3, so that'd be like 0.3 standard deviation units, and we defined the triviality threshold as 1/3 of that. If we wanted to have really strong evidence for a greater than trivial effect, that needs to have more sample size required than this. This will allow you to get a p less than 0.05 for a trivial effect. And this doesn't allow that. So this has to be bigger. But when you run the design, what did we find? Your average sample size is only 113. So you're actually bringing in a more stringent assessment, but because you're doing a lot of looks at the data, your sample size is a half of what the frequentist design needed as a fixed sample size design if you were to use 234 as your sample size. Now, the calculation for the minimum N for precision was 60. We're going to go ahead and set a maximum N of way out there at 750. So we run our Bayesian simulation, which means you're simulating a different effect delta for each trial. Our goal of Bayes is to uncover the hidden truth that generated the data. Our goal of Bayes is not to say if delta were something, we have just been surprised by our data. Bayes couldn't care less about that. So when you're doing your simulation, you have to have a universe of delta to simulate from. And so this universe was chosen to be disadvantageous to the Bayesian procedure that I'm using. So how does that play out? So I'm using a normal distribution for delta, the prior distribution, and in the way that delta was drawn for the simulations, the way the deltas are generated is such that the probability of a really big treatment effect is very small. The probability the treatment effect is harmful, so it's less than minus 0.75, is also 0.025. So we have a very small probability of a very large treatment effect. But the way that we're analyzing the data is we're more favorable to a large treatment effect. So instead of 0.025, we're using 0.2. So that's quite a heavier tail out there. Much more likelihood of a big treatment effect in the way we're analyzing the data. That's the analysis prior. This is the simulation, or what's called sometimes the sampling prior.
And is that centered at 0, your sample
Yeah.
size?
Yeah. We're going to do 10,000 trials, sequential assessment with unlimited looks. So how often did we stop at any time with a conclusion of inefficacy? We stopped, out of 10,000 trials, we stopped more than half the time with an early conclusion of inefficacy, which includes futility with regard to efficacy. Now that is a huge savings. We didn't go to 234, and we definitely didn't go to 750, which we could have gone to. So we stopped, and the average, after 62 patients, we had strong evidence for efficacy that's less than a trivial amount. How often are we correct? This is the bottom line. Is the Bayesian procedure correct in the decisions it makes? So out of the 5184 times we stopped early, concluding inefficacy, we were correct 5,020 times. So 50/20 out of 5184 is 0.97. So when we stop early and conclude inefficacy, we're right 97% of the time. What about stopping anytime for similarity? This is harder to demonstrate. So you're going to stop later, and you're only stopping 634 trials out of 10,000 with the ability to make a conclusion that the two treatments are similar in their outcomes within plus or minus 1/2 MCID. So we stopped 634 at an average sample size of 423. How often were we correct? 607 out of 634, 0.96. Now, how often did the worst thing happen? We have limited that. Only 172 trials went to the maximum sample size. This is what happens most of the time in a clinical trial designed with a fixed sample size using a frequentist p-value. That's the most common occurrence, is you'll come up with an inconsequential equivocal result. Now, what makes this happen? This stands to reason what happened. What makes you unable to say what the treatment does is that the true effects of those 172, those clinical trials were generated when the true effect was right at the threshold. So your threshold of triviality was a 0.1 for gamma, and the median of all the deltas when this happened was exactly 0.1, and with a very narrow spread away from 0.1. So you're right at the threshold, so you really have maximum indecision when you're right at the triviality threshold. It all makes sense. But we've avoided this happening very often. And what about what we hope for? Stop at any time you're greater than 60 patients with a conclusion of non-trivial efficacy. So we stopped early 4,010 times. So that means we have a high probability that delta is greater than 0 and a 0.85 or greater that delta is greater than gamma. The average time at which we stopped was a sample size of 102. So we have adequate precision and we have a high probability of efficacy. And how accurate were the decisions? Well, in terms of how often you were correct that the treatment is better than trivial, you were correct 3643 out of 4010. That's 91% of the time. Now remember, we only demanded the probability of capital delta being greater than gamma to be 0.85 or greater. If you wanted to make that 0.85 equal to 0.9, this would go up. But in terms of being correct, in terms of the direction, we are correct 98% of the time. So when we stopped early with a conclusion of non-trivial efficacy, with regard to efficacy, we're, in other words, the correct direction of effect. We're right 98% of the time. Now, what is especially important to note, these are already really good. This is when you have put your analysis at a disadvantage. So even though your posterior probabilities are going to shift a little bit because of a disagreement in the prior, what it does to the accuracy of your decisions is not very much. So in spite of a mismatch in priors that works against the procedure, our accuracy is extremely high. And that's all we care about. If you market a drug, what's the chance that it doesn't work? FDA cares about that a lot. So if you have a 0.98 chance that it works and your prior is agreed upon, the chance that you're wrong is 0.02. If you market a drug because the p-value is less than 0.05, there's no way under the sun to calculate the probability that you just made the wrong decision. That is unobtainable from the frequentist paradigm. So, in summary, if you need to have disciplined choices. We can't have a game where we keep modifying the MCID. We need to have good outcomes. And if we have a margin for triviality of effect, will help the clinical interpretation and the performance of the study. And we will abandon futile studies before all the money's spent. It's the best world to not have a sample size calculation. So how do physicists do experiments? They don't do sample size calculations. They experiment until they have the answer. That's called a scientific approach. And it's funny that in traditional statistics, we don't do what physicists do. Physicists know what they're doing when it comes to experiments. Sequential designs can easily have multiple explicit clinical goals in the Bayesian framework. They can greatly speed up research, and you can get impressive Bayesian operating characteristics even with conflicting priors. So I want to thank everyone for bearing me out and for being here today, and thanks to you online also. So I look forward to some more discussion and questions.
That's great, Frank, very informative.
Tried to be.
Yeah, questions.
Is it sort of the focus of the work that you're doing with the, I mean, because, this is proposing a huge shift to a paradigm that exists with that has an enormous budget and so is this the nature of what you're working on when you have this work that you're doing with the FDA? Is this what you're working on?
Yeah, so what motivated me writing this chapter is every time a statistician would express a concern about a Bayesian part of the analysis, it could be an FDA statistician, could be somebody else, somebody on Twitter. I'll create another set of arguments or examples. So this is a response to all the things that have been discussed that I've been part of for the last 6 or 7 years. And so it's very much responsive to that. And so like we have a working group at FDA on multiplicity. What does multiplicity really mean? So that made me write a section on multiplicity elsewhere in the course notes. And every time a topic comes up, I try to think of what are some examples, what are other ways to think about it.
I think it's really hard to get people who have budgets to, people want to know what the budget's gonna be.
Yeah.
It's really, really difficult to get people away from that because we have fixed budget, we only have so much money, we have
Yeah.
limited resources. And I think as you pointed out very early on, like part of the strength of all you're talking about is stopping early. So you say,
Yeah.
that's really like the biggest, like that's clearly like a selling point to this approach. Yeah, it's super interesting.
So to me, it's an easy sell in the pharma industry because they have budgetary control. It's really hard with NIH. So NIH is the one holding us back because they have fixed budgets. So the line of attack and where what you said is so important is you hire staff, you train people at sites, you gear up everything. And how are you going to hire people and then let them go quickly? So where we need to push is things like platform trials. So when you have a platform, when one part of the study shows futility, you move to another part of the same study. So platform trials are like a magical recipe when you can do them because of the issues you raised.
So how many Bayesian trials are underway or?
Well, I work with a group at Imperial College London called the Orbita Group, and they're converting all their trials to Bayes. And we did one trial that was very successful, published in the Lancet, and it was totally Bayes. And it was a watershed event when the reviewer wrote back that you had one supplemental analysis that was frequentist that needs to be taken out because this is a Bayesian paper. I knew we were on the road.
But what about on this side of the pond?
It's, you know, when we did the Active 6 platform trial for repurposed drugs in COVID-19, we used Bayesian methods for the monitoring, all the interim monitoring, and we wanted to use it for the main analysis, and NIH vetoed it. That was a great disappointment, but we ended up using Bayesian in one of the main papers. It was one about ivermectin, where we used the Bayesian result to better interpret the results of the trial. So we used it in a mixed way in our COVID-19 series of trials.
I have a hard time understanding the difference between the MCID and the trivial effects.
Yeah.
Talk a bit more about that.
Yeah, what got me on that line of thinking is if you would like to show Bayesian evidence that the effect is greater than the MCID, but you can't do that. The reason is most of the priors we use are like centered at 0 and they're symmetric. And so if your observed effect in the data was right at the MCID, the posterior probability being greater than that is 1/2. And that would really disappoint the clinicians. So it's very hard to show a probability of 0.9 of being above the MCID when all you got in the treatment effect was the MCID. So you don't want to call that a failure. So you can't calculate something right at that margin. But then you ask, what's the smallest effect that could possibly mean anything? It doesn't have to be clinically effective. You know, it doesn't make the patient return to work. They have a lot of back pain. They can't go back to work, but they tell you their back pain is noticeably better. So that's what we're talking about, is like the minimum visible effect. And when you, if that's like 1/2 the MCID and the result came out at the MCID, the probability being greater than 1/2 the MCID could be large. It's kind of a backwards way to think, but it's practical.
Hey, Frank, what if you've done your analysis in the frequentist way and you have your confidence intervals with, you know, alpha of 0.05? Would it make sense to interpret the results after post hoc choosing MCID? I mean, just thinking it through, what if I had this kind of MCID? How do I interpret this result? Or would it make any sense to do?
Well, you know, there's a lot written about how to interpret confidence intervals, and nobody has ever really solved that problem. But the reason that we dwell in the posterior probability space is that you give it the interval and it outputs the probability. And whenever you're giving something a probability and it outputs the interval, like a confidence interval, you lose control of the clinical interval you're interested in. And so that's only part of the story, because you're asking also about the MCID specifically. How are you going to conclude that the treatment is meets your expectations with regard to the MCID? So I think that's actually a difficult question, and it's not fully worked out yet. I mean, you would like the MCID to be in the Bayesian uncertainty interval at least. Or it could be, you know, your Bayesian uncertainty interval could be all bigger than the MCID. That would be a dream situation. You know, you went way beyond the MCID. So it's not a very simple answer to that, and I'd like to work on that some more.
You mentioned the NIH is vetoed a lot of use of Bayesian trial design. Are any of their concerns or reasons for vetoing it founded, or is it just bias towards?
It's really habit. And so the term that I use for this in general is just like we have trouble getting people to use ordinal regression. So Brian and I and others have been on a crusade to overcome that. But the main reason people don't use ordinal regression more often is they're not familiar with it, because most graduate programs, unlike ours, don't teach it. And so I say the term unfamiliarity breeds contempt. And it's like that with Bayesian methods. So I talk to people who have reservations about Bayes. First question I ask them, how many Bayesian courses did you get in graduate school? The average is about 0.5. And a lot of people I ask that to, including most of the students at UNC today, the average student at UNC, when they get a PhD, have 0 Bayesian courses. And that should never, ever happen in this day and age. And we don't let that happen here. So a lot of it's just unfamiliarity. And then number 2 is people have been drilled into thinking you need to preserve alpha. So one of the most famous people at FDA, who's a non-statistician, got up at a big meeting one day and said a p-value is the probability that the null hypothesis is false. And he couldn't be more wrong. But he said that's how he understands it, and nobody could ever shake him. I went to his office and I sat for 2.5 hours going through all of what probabilities mean. And at the end of the meeting, he said the p-value is the probability null hypothesis is false. And so, it didn't matter. He was unshakable. So, and then people use the word error, thinking that alpha is the probability of making an error. It's nothing to do with that. But we use the word error, it gives it some kind of reverence. So one of my main teaching goals is to shake that reverence off of alpha. It never accomplished what the originators of it thought it accomplished.
So I heard from somewhere that folks in the pharmaceutical companies are really reluctant to stop early for fatality because there may be only few ongoing trials and they need to have some successful drug for to set a complaint. So what if actually they really need to stop the trial for fatality, but they just ignore it and go ahead? And if at the end a miracle happens, like to what degree can we really believe that the drug is actually effective?
Yeah, I mean, miracles can happen. You can see data turn around, but they happen so infrequently. And when you're looking at your expected gain from information, you have to play the odds. You can't play for miracles. You have to play the odds in the long run. There'll be exceptions always. You can have a strategy that's going to lose you one night in Las Vegas. Actually, most nights in Las Vegas, but you have to play the odds and go with the long run. And you also run a whole lot of simulations and show the consumers exactly what happens in all those simulations. And they can point to, here's what they would like to have happen. And you tell them, well, that happened in 3 trials out of 10,000. They might say, they think they can get an exception, but it's not going to be a good play.
I can follow up on that. I feel like there's looking at this, I imagine someone very diligently holding the coin purse and trying to spend the money very wisely. But I feel like for better or for worse, the way I see some, not all, but some of the NIH funded projects, they're more they have that kind of startup culture where you get the money and then it become a game of, oh, I can spend it, so why not?
Yeah.
You see what I'm saying? So
yeah.
I feel like there is a disconnect between the philosophy here versus maybe some of these real world projects are run, at least given how they're currently
Yeah, there's a lot of psychology
Yeah, yeah.
and a lot of regret and a lot of minimizing expected regret. There's a whole field of psychology that studies these kind of decisions. But one time at Vanderbilt, about 20 years ago, we assembled a panel of medical ethicists at Vanderbilt and charged them with the question is, here is a study that was designed using a faulty analysis where they took an accepted scale and dichotomized it and ruined the power of the study. Is this study ethical to conduct? And they unanimously voted that it was unethical to run that study. So when you have, that was a different situation. But the reason they said it was unethical is that patients have a valid expectation of contributing to science. If what you're doing, like there's a lot of clinical trials that are started, that if you did a futility analysis before any data was collected, you would say it's already futile before you have any data. Because you have a flaw in your outcome variable, your ultimate sample size is way too small. And so patients have a right to expect they're contributing to science to undergo risks and inconvenience. And I think it's an ethical problem if you keep a futile study going.
What if it's not something that's, what if it's something that represents minimal risk? Well, I'm not thinking not in the trial context anymore, I'm thinking like, file specimen, running samples, doing the mathematics, that kind of study design.
I think it's worth getting patient input about this. The problem is we're never honest enough when we talk to patients. We ask them, we do studies about how well they understand informed consent, but we don't really talk about these things. And we don't tell them the truth about the majority of clinical trials are equivocal. And the patients who were in the studies contributed nothing. Zero. We don't say that to patients. I think if you want to have that discussion and make it very honest, and they decide.
I mean, like, at the end of the day, like, it's hard enough to get people to participate. I'm not saying you should lie to people, of course. But, like, and they talk about, like, if you're doing an informed consent, it needs to be at, like the 6th grade level, so that people can understand it. I think that some of these concepts that you're talking about are intellectual and challenging, and I think that that there's a tension there, that it's not so easy to implement that, what you're describing.
But Jonathan, if you were a patient enrolling in a study and you saw a sentence that said, this study is designed so that if there's strong evidence that we're not going to be able to find a benefit of the treatment being studied, we will stop.
That we're not going to be able to.
Yeah, there's a high probability that we're not going to be able to show that one treatment is better than another. We will stop the study early and you'll be released from the study. Wouldn't that be a nice thing to read?
That would be nice.
We don't have that now.
Yeah. I mean, informed.
When you stop early, your point estimates are biased, right? So if you stop early for success, you're gonna get too high of a point estimate. When you stop early for failure, you get too low of a point estimate. Is that correct?
That's strictly a property of frequentist methods.
Because you don't believe there's a single point estimate. But if you compare it to your delta, you generated the data.
So when you get the posterior mean of delta, it's perfectly calibrated no matter when you stop.
It is.
Yeah, it's automatically pulled back by the prior in proportion to how early you stopped. I've got an example of that in the course notes.
I understand that. Because to me, it seems like you're going back and forth, you're close to the border, then you're gonna stop when it jumps over, and then that's gonna naturally make it a little on the bias.
Yeah, it's only going to be seen as a problem if you have second thoughts about your prior. Do we have any online questions?
Not right now.
Okay.
I would get like follow-ups, like if somebody wants to do a follow-up study or meta analysis.
Yeah, right. Like what is really the effect size? Yeah, stop. We've made the dichotomous decision that this is effective, but So our point estimate, higher.
So anybody hasn't asked a question yet?
Anyone has a what?
Anybody that hasn't asked a question yet who wants to ask a question? Anybody wants to ask a repeat question?
Very thoughtful.
That's the goal.
Cool. I think one thing that's cool, and maybe I should have thought of this 1,000 years ago, but the idea of doing the simulation where you're not, we don't have a single point, you don't have a single effect size, you have a distribution of effect sizes. That's sort of like something that I haven't really done before. Truthfully, not even thought about. So
It's something everybody should look at the code for. When you look at the R code for that versus the frequencies, the whole paradigm becomes clear. The code tells all. I got into an argument in a restaurant once, I think it was in Richmond, Virginia, where I was arguing with a statistician that said there's multiplicity with Bayesian sequential testing. And I said, well, if you believe that's true, you have to identify the error in my code. And I challenged him to do that. He was unwilling to take up the challenge. But we almost got thrown out of the restaurant. We got super loud in this argument.
You need to tell me later.
Yeah, I can't remember who it was. It wasn't you. It wasn't you, Bob.
Yeah.
Yes, we are. Thanks.
Okay, thanks, everybody.
Thanks for putting that on Twitter. I read
Hey.
your blog on 7.
Oh, good.
I'm just like that. So.
Thank you.